Hey, good afternoon. It's an honor to introduce Dr. Patricia Dahia for our weekly workshops seminar. I believe Dr. Dahia and I came to San Antonio around the same time, early 2000. from the same location in New England. Dr. Dahia is an MD PhD, so she's dually trained. She received her MD from Federal University of Paraíba in Brazil, and her PhD in University of Sao Paulo in Brazil. Then she went to London and did her postdoc in molecular endocrinology, and then on to Harvard, Dana-Farber Cancer Institute, and then did another postdoc in cancer biology. So we are both endocrinologists, another thing we share, we have in common. And Dr. Dahia is really a world-renowned expert in the genetics of pheochromocytoma and paragangliomas, a disease that can be devastating, and her research is really important to unravel what is behind these disorders. And she's also developing, she's always had an interest, but now she has a leadership role in translational research and is on the leadership of our CTSA. So that's another very important role that she has. She is very well funded. She has grants from NIH and CIPRIT, and her research is published on the top journals. So we're really lucky to have her in San Antonio to be part of our faculty and training grant, and having her come and teach us about her research. Thank you, Dr. Dahia.
Thanks so much, Nick.
Is this working? Yeah? Yeah? Everybody hears me? Okay, it's hard to tell. Thanks so much for the kind words. It's a pleasure to be here. So some of what I'm going to talk about today, you have already heard from Subu's words. So Subu is a postdoc in my lab, and he's a fortunate T32 postdoc trainee. And a lot of what I'm going to talk about is the product of his daily labor. So I wanted to give him full credit right now. And of course, I'll give it again at the end, but I just wanted to make sure that this is fully acknowledged. So I will talk about different aspects of Tmem127. This has been a major part of what my lab has been dedicating to. And as Nick was talking about my background, for many years I was only working on the cancer genetics portion of endocrinology. So I've been interested in pheochromocytomas for a long time, initially as a clinician following patients, and then later on only in the lab as I've continued to be. But because my training was initially as a clinician and then the basic research came afterwards, the focus of the research is really very translational. It's disease-oriented. I like the idea that we have a question in the bedside that we can bring to the lab and try to address it and hopefully do something there that can be then taken back to patients and disease models. But Tmem127 was a gene that came along to us, and I'll tell you a little bit about it, how it came about, and that took me back to my origins as endocrinologist and made me study a lot of things that I hadn't seen in a long time. So you will see how. So how we came to Tmem127. So many years ago, we found a family that had many individuals with pheochromocytoma. It happened to be a very large sibship with 12 individuals, 12 siblings, 6 of which had pheochromocytoma. And they didn't fit into any of the only 3 syndromes that were known at the time to have familial pheochromocytoma. So we were intrigued by it and started to imagine ways to figure out what the gene was that was causing the disease in that particular family. So let me tell you what pheochromocytomas are. So these are neuroendocrine derived tumors. They arise from the adrenal medulla for the most part. So they are called pheochromocytomas because it's a long story how the name came about, but pheochromocytomas and their related paragangliomas are tumors that derive from chromatin cells, which are cells that make or secrete catecholamines, noradrenaline and adrenaline. For the most part, these are benign tumors, but they occasionally can be malignant, and it's very hard to distinguish a benign from a malignant tumor unless you have metastasis. So we had this family, and we studied them for a long time. We excluded parts of the genome as being the culprit and eventually found the mutation that caused the disease. So from this original discovery, many other patients were found to have mutations in this gene, and Tmem127, and the name is given because it's a transmembrane protein, and the number 127 is just because it was the 127th of genes of unknown function that had transmembrane protein. So it codes for a transmembrane protein of unknown function. It doesn't have any known motifs that we could classify it into belonging to certain classes of disease. So we've been studying the gene ever since. So as you can see here, this is the linear structure of a 238 amino acid protein. It has 3 predicted transmembrane domains. And these are all the different mutations that have been found from our initial reporting that almost 8 years ago now. So the mutations tend to be loss of function or truncating types. And they are part of autosomal dominant disease, so they're all germline. Individuals are born with the mutation. And in humans, the only phenotype that is associated with these mutations is pheochromocytomas, paragangliomas, or more rarely renal carcinoma. So it's only tumor phenotypes. It's an official cancer gene in the Cosmic Catalog. And what we know about it is that it's very conserved across species, but only up until the lowest vertebrate, not below vertebrates. So different from other transmembrane proteins. We don't see it in worms, or we don't see it in flies, and we don't see it in yeast. It's ubiquitously expressed, and as I mentioned before, it doesn't belong to any known functional class. So we started off our journey to try and understand what Tmem127 does through two different approaches. One was based on the fact that it was a transmembrane protein. We wanted to see structure where it distributed within the cell. And the other part was based on some microarray expression data from tumors that had mutations versus tumors that didn't, trying to figure out what kind of pathways might be disrupted. So I'll talk about the first part. So we found that expression of Tmem127 tagged with different types of tags would give us a similar pattern of punctate appearance in the cytoplasm suggestive of an endomembrane domain. So we found that it actually colocalizes with multiple endomembrane domains, early endosome, recycling endosome, late endosome that is not shown here, Golgi complex, lysosome. And endogenous Tmem127, in fact, is only seen in membrane-enriched fractions, but not in soluble cytosolic fraction. What we also learned was that when you have a mutation that is seen in patients, and if you model that mutation by expressing it in cells, these mutants tend to have a diffuse appearance, and that is compared to the punctate appearance of the wild type, giving us a hint that being part of the membrane is important for its function and its tumor suppressor role. So more recently, Shahida Flores, a graduate student in the lab, has found something really interesting based on a mutation that was found in a patient. So she found that having a mutation in a certain location of the gene, which would not be predicted to be a transmembrane domain, would give a diffuse appearance. I don't think you can see it very well, but this is diffuse as opposed to the punctate appearance. And that is not the typical thing that we see in missense mutations that are outside transmembrane domains. So that took a, so we also had a mutant model where the N-terminal portion of the gene, which does not have a transmembrane domain theoretically, deleted. So this is the N deletion mutant here. And you can see that both have a diffuse appearance. So as it turns out, there is a prediction of another transmembrane domain here. So this is something that we are exploring right now, but what it turned out is that based on a single patient that had a mutation in a very strategic location, we were able to see that the prediction model for this transmembrane protein was incomplete, and perhaps Tmem127 is in fact a tetradomain spanner rather than a three-spanner transmembrane protein. And that may have implications for what we will eventually find as its function. So the other part, so this was the kind of a distribution, colocalization part. The other side of the story was that we wanted, yeah, that was a HeLa or 293, so we use either. So just to remind everybody, Tmem127 is ubiquitously expressed, so we don't have existing human-derived pheochromocytoma cell lines, so for the most part, we use any of the common cell lines for many of the experiments that we do. So the other side that we tried to explore was the fact that we had several pheochromocytomas from patients that had Tmem127 mutation, and patients that did not have that mutation had different pheochromocytoma-related mutations. So in a microarray-based study, we found two subtypes of pheochromocytomas, and we found that Tmem127 tumors invariably related to those associated with increased kinase signaling and protein translational pathways. So we used that to try and get an initial hint on what Tmem127 might do. So this is just a combination of the tumors that belong to this pathway and some of the genes that were known to represent the pathway, such as RET kinase receptor or NF1, which is an inhibitor of the RAS pathway. And as you can see here, the mTOR pathway is a common converging point from all those different kinase signaling pathways. So we tested whether knocking down Tmem127 would increase the signaling through mTORC1 pathway, and in fact it did. So you can see here, so those were two different guides that we used at the time versus the control cell, and that happened in conditions of serum stimulation, amino acid stimulation, which are known activators of mTORC1. And if we, in contrast, overexpressed Tmem127, we would have a decrease in TORC1 signaling. On top of this, we were able to test mutants that had Tmem127 loss of function type mutation, and these are pheochromocytoma lysates, and compare with those that didn't. And again, we saw that mTORC1 signaling was activated in this tumor, suggesting that Tmem127 had deficiency or mutations, loss of function type mutations would activate mTORC1 pathway. And I wanted to remind you that mTORC1 at about that time was being intensely investigated in terms of its signaling and the role of lysosome in its response to amino acid and nutrient stimulation. So just to briefly revise, in the absence of amino acids, mTORC1 is located in some very still poorly characterized portion of the cytoplasm. And in the presence of nutrients such as amino acid, mTORC1 is transported through a GTPase called RAG to a lysosomal surface location where it can be activated by RHEB, R-H-E-B, which is another GTPase that is the terminal activator of mTORC1 signaling. So for that to happen, there has to be a conjunction of events in binding. So a VATPase complex subunit is a critical component of this lysosomal complex. Regulator, which is a pentamer where proteins called mTOR 1 through 5 are also required, as well as the reg-GTPase. So regulator bound to VATPase functions as an activator of these GTPase, which then captures mTOR and brings it to the lysosome where it's going to be associated. And why am I saying all this? You will see in a minute. So we had fibroblasts from the knockout mouse model that I'm going to talk about, but this was just done with primary fibroblasts that we immortalized in the lab. And we, as you can see here, if you compare a knockout fibroblast that doesn't have TMEM with one that has been rescued with Tmem127 expression, there is increased mTOR signaling in the knockout cells, which is pretty much ablated by the re-expression of Tmem127. We also found, and this is work done by Ilun Deng, that was part of her thesis here that was completed recently. So what Ilun found also is that TMEM has a very similar dynamic of association with the lysosome, which we're showing here as co-localization with the LAMP2 lysosomal marker. And they both increase after a few minutes of amino acid stimulation and then fall afterwards. So that told us that TMEM tend to go to the same lysosomal space where mTOR goes to be activated by nutrients. And in fact, we thought that this might be happening because TMEM binds to one of the regulator components, both ectopically and endogenously. And the effects of that are increased by the presence of amino acids. And when TMEM is expressed, it reduces the ability of components of the complex to bind to each other. So we think that this is potentially one of the mechanisms through which the presence of Tmem127 reduces mTORC1 signaling at the lysosomal level. So this is kind of the model that we came up with. So interestingly, Tmem127's effects are seen in the presence of amino acids, not in its absence. So it's not an inhibitor such as TSC, which functions in the minus on the off mode of mTORC1, but rather on the on mode, whereby we believe that it attenuates and modulates the signal and the duration of the signal by its absence or presence. So in summary, just this portion of the work, this is all earlier work. Tmem127 is a tumor suppressor, bona fide tumor suppressor gene. It's ubiquitously expressed. Its presence located into endomembranes, endosome, and lysosome. Its loss of function leads in humans to increased kinase and mTORC1 upregulation, and it associates with the lysosomal complex that activates mTORC1 in response to nutrients. So with all that, we wanted to get a better sense of how exactly it is doing this and how it works. And we developed a mouse model that was a conditional model, but with the hypothesis that maybe Tmem127 being ubiquitously expressed and being a cancer gene would lead to embryonic lethality. We crossed it with a generic CRE, a CMV CRE, to test the hypothesis that Tmem127 might be lethal, its absence might be lethal to an organism. And in fact, we found that it was not the case, although after at the P0 or the day of birth, we have a slight reduction in the number of knockouts compared to wild types. But for the most part, it's roughly a Mendelian distribution. So we had enough of the nulls that we could work with them. And another thing that we noticed was that post-birth, but not pre-birth, there was a decrease in the body weight of these embryos, and that happened in both genders. And this is a part that you've seen Subbu present a number of times. So we found that the surviving knockouts were smaller, from birth, and then they continued to be smaller throughout their lives. And what was more remarkable there was the fact that they were predominantly reduced in their fat mass compared to their lean mass. So they were overall small in everything, but they had a more marked decrease on the fat mass compared to the lean mass. And this is just a representative example of how much smaller their inguinal fat was compared to the wild type. And we also found that in terms of their fat, both white and brown fat, there was a reduction in the size of the cell rather than the number of cells. So at this point, I was very curious because our hypothesis was that this being a pheochromocytoma gene, a tumor suppressor gene, its absence would make the mice prone to developing pheochromocytomas. So we tested catecholamines both in the plasma and the adrenals, and the ratios of norepinephrine to epinephrine. And we had no evidence, biochemical or histological, that there were pheochromocytomas in these mice. And we have been able now to follow them for their survivorship. So we know that it was not a late event only. They simply did not develop pheochromocytomas, and we haven't seen any. So one thing to explain their phenotype is, could they be lipodystrophic then? And we measure their glucose and insulin levels, and we found that the glucose, fasting glucose, tend to be lower on the knockout, and their insulin was substantially lower. So were they then the opposite, insulin sensitive? So we started testing these mice systematically. And remember, this is a straight knockout, TMEM knockout. So we didn't see much of a difference between the wild type and the knockout when they are young and up to 6 months old. But then as they get older, we see a split in their glucose tolerance curve and the insulin tolerance curve, suggesting that they have better glucose-insulin tolerance. And if you give them a glucose stimulation, then the insulin levels of the knockout don't rise quite as much as the wild type. And if they are subjected to a pyruvate tolerance test, then they also perform better, suggesting that they have reduced gluconeogenesis. So our conclusions at this point is that the adult TMEM knockout mice are insulin sensitive. And because this happens and it becomes more accentuated as they age, they were protecting, the absence of TMEM was protecting the mice from the age-induced insulin resistance. So the next question we had was, what if we induce obesity? Will these mice be protected from obesity as well? So they were subjected to a course of 16 weeks on a 60% high-fat diet. And they actually put on weight in relative terms because they started off smaller. They put on even more weight than the wild-type mice. And their fat mass became the same as compared to the original starting point. Their lean mass stayed smaller, and they were not really resistant to the obesity effects of a high-fat diet. The cells got bigger. And they had a glucose tolerance curve that was quite similar and overlapping, but the insulin tolerance curve was still much better than the one of the wild type, suggesting that they remained somewhat insulin sensitive despite the high-fat diet exposure. So then we wanted to understand why they had that lean phenotype, that protective phenotype. So we looked at food consumption, imagining that maybe they ate less than the wild type, but we didn't find that. We also considered that maybe they were physically very active and measured their locomotor activity, and we found it to be significantly lower. So they were moving less, and I'll come back to that at some point, both at night and day. We also considered that maybe they were thermogenic, so they were burning their fat at a greater rate. And we didn't find in any of the fat depots that they had higher levels of thermogenic markers such as PRDM16, UCP1, adrenergic beta 3, and also they didn't have higher, more elevated body core temperature. So it didn't look like they were obviously or overtly thermogenic. And we also consider that they may be just having higher fat oxidation, and they had respiratory quotient that is measured as an overall body assessment of fat oxidation, or usage of fat as the main caloric substrate, and they were no different from the wild type. We did see that on high-fat diet, both wild type and knockout decreased somewhat their respiratory quotient as expected, because you're providing more fat as substrate, but they didn't differ from each other. And in fat tissue, genes that were involved in both lipolysis and fatty acid oxidation were not increased in these mice. So we thought that could they be, instead of burning too much fat, making less fat? So we looked at that next. And here I'm comparing liver, muscle, and fat tissue from wild type and knockout mouse. And we didn't see in fat tissue that was a decrease in fatty acid synthesis. In fact, they were making quite a bit of it. Muscle didn't show much of a difference or a decrease, but in the liver, we saw that the total ACC and fatty acid synthase levels were decreased at both the protein level and at the mRNA level, as well as markers of storage. In addition, to support the idea that the liver was making less fat, we had significantly less liver triglycerides in the knockout mouse. So what happened if we analyze the liver from the high-fat diet subjected knockout mice? We saw that their liver at the end of the high-fat diet period was significantly smaller than the wild type, so it had increased from the chow, but it was still smaller, both at absolute levels and relative levels. And histologically, knockout mice had less accumulation of fat in the liver compared to the wild type. When we looked at the mRNA levels of the same markers that I just showed you on the chow mice, we found that their liver expression level of those genes that are involved in fatty acid synthesis and storage were still decreased. So the high-fat exposure did not make the fatty acid synthesis process go through. So that also happened at the protein level. So our idea at this point is that these mice have a decreased ability or capacity to synthesize fat in the liver and to store the little fat that they make. And so that could potentially be one of the core defects that the mice had that would make them potentially explain their low adiposity and the insulin sensitivity. So just to remind you then, they had low ability to make liver lipogenesis and low locomotor activity. So we wanted to start testing some hypotheses. So the first one that we wanted to make sure that we either confirmed or excluded was the mTORC1 activation model. So as you recall from the beginning of the talk, we found that there is a connection, negative correlation between TMEM and mTORC1 signaling. So one of the models of mTORC1 activation in mice is a liver model of TSC1 knockout. And these mice have some features that are quite similar to what we were seeing in the TMEM knockout. So these are mice characterized by reduced locomotor activity. They have low core body temperature. They had decreased liver lipogenesis. And as an mTORC1 defect activation, one might expect that those were features that could be reversed by rapamycin, that is an mTORC1 inhibitor. So we tested that. Now, in this model, even though there is high mTORC1, the idea is that mTORC1 will inhibit by a negative feedback AKT, which is the primary stimulator of fatty acid synthesis in the liver. So the reduced de novo lipogenesis in this model is attributed to reduced AKT levels. And what they found in these mice was that the high mTORC1 activity stimulated PGC1 alpha, which in turn would induce FGF21. And the FGF21 was the ultimate reason why these mice had decreased locomotor activity and low body temperature. And that was shown. This is a paper by the Michael Hall's group in PNAS. And they showed very neatly that if they knocked down FGF21 in these mice, they would completely reverse the phenotype of low locomotor activity and body temperature. So we wanted to test the same thing in our mice. So could it be due to AKT suppression? Could it be due to an FGF21 induction? And would RAPA be able to reverse that? So we found that so our mice had increased phosphorylation of S6K as an mTORC1 activation would have produced, but they did not have suppression of phospho-AKT. So that was different. Also, these mice had no increase or induction of FGF21 either in the fed or in the fasted state. And finally, the triglyceride reduction in the liver was not reversed by rapamycin. So whatever we were seeing was not due to mTORC1 activation. So then mTORC1 has a very complex and intertwined relationship with mTORC2. So TORC1 can inhibit through this process TORC2, but TORC2 through its phosphorylation of AKT can stimulate TORC1. So very complicated to detangle these two. But we thought that maybe we were looking at the wrong TORC complex. And potentially Tmem127 might be inhibiting mTORC2, so its absence could be stimulating mTORC2 activity, and that could potentially explain the insulin sensitivity of this model. So we wanted to test that by looking at what rapamycin could do to it. Since rapamycin is a known, well-established mTORC1 inhibitor, it's also known that chronic use of rapamycin will eventually inhibit mTORC2 as well. And another bonus there is that chronic use of rapamycin, because of this effect on mTORC2, this is believed, would so patients that are under chronic use of rapamycin or mouse models of chronic use of rapamycin have increased glucose intolerance. So the same setup where we measured liver triglyceride was used here. So we had the mice be injected with IP RAPA for 4 weeks, and at the end we collected the mice and compared with vehicle treated. So while the expected glucose intolerance happened in the wild-type mice, we did not see the same thing with the RAPA-treated knockout, as you can see here. And the same thing with the insulin tolerance test. So the insulin levels continued to be low. They increased a little bit, but not to the same extent that the wild-type. So these data suggested that rapamycin use did not cause glucose intolerance in the Tmem minus mice compared to the wild type. So we thought that maybe these mice were behaving that way because they had too much mTORC2 activity, and the amount of rapamycin that we were giving was not enough to completely suppress it. And we tested it by looking at the mTORC and Richter complexes, which is the core and the unique components of the mTORC2 complex, and we measured it by IP in the 3 models that we were studying where we saw that the Tmem knockout mice were insulin sensitive when compared to the wild type. And those were adult, so mice as they aged, high-fat diet exposed mice, and the rapamycin-treated mice. And in all 3 models, we found significantly higher levels of Richter mTOR in the knockout mouse compared to the wild type, suggesting then that these mice have increased mTORC2 complex abundance, and that might translate into more mTORC2 activation, which could potentially explain their insulin-sensitive status. Now, we know that Tmem127 expression is ubiquitous, and here we're just showing some both in mouse and the human data. This is from the GTX consortium, where there's data from thousands of humans that in all sorts of tissues. I'm just showing here the tissues that might be relevant for the metabolic analysis that we were doing. And we saw that both in mice, Tmem127 is expressed at about similar levels. And I just wanted to highlight the fact here that muscle appears to have a lot more, but as it turns out, we were having trouble distinguishing wild type from knockout using just plain whole cell lysates. When we fractionated the lysates, we found out that there is a cytosolic protein that binds to the antibody with the same size, and it was throwing us off. So this is the actual muscle expression of TMM. So it's pretty similar to liver and fat. And so one question that we had was, okay, so TMM is present everywhere, so does its expression change in a way that would make it a good candidate for a metabolically responsive gene or protein? And what we found was that by looking at liver, we found that TMEM expression at the mRNA level increases with age, increases with high-fat diet, and reduces back to normal when the high-fat diet is switched back to a chow diet. And it's higher in mice that are the DB mice compared to heterozygous. So what these show in common is that TMEM tends to be more abundant in conditions where there is insulin resistance, regardless of the diet type, because the DB mice are obese and insulin resistant even on a chow diet, right? So that was interesting, and it fit in with the data that we had. So the model that we're still trying to refine is that TMEM potentially inhibits mTORC2. Now, mTORC, and that would promote insulin signaling and sensitivity. The other important point there is that Tmem127 somehow induces de novo lipogenesis in the liver. And we don't know if this is by a separate mechanism than mTORC. mTORC2 is not known to have specific effects on reducing de novo lipogenesis in a way that would justify everything being caused by mTORC2. But one possibility is that the low levels of insulin, circulating insulin, that these mice have in virtue of their insulin sensitivity, high insulin sensitivity, maybe there's less insulin around, so maybe part of the reason why they have low adiposity is that they don't have too much lipogenic stimulus in the fat. Or there may be something else coming out of this liver that is not being so stimulating for adipocytes. So we're still working on refining the model. But with the thought at this point that we had potentially a primary liver defect based on the straight knockout, we made a liver-specific knockout by mating our flox mice with ALB-CRE. And these CRE mice were kindly provided to us by Luke Norton. And we accompanied these mice and looked at their body weight, which was how we got started on this, and also their body composition. So what was interesting in this group of mice was that we found that they were two subgroups. There was a 50% penetrance of a lower body weight, lower fat mass and low lean mass. And the other group, the other mice seemed to be similar to the flox mice in terms of their body weight and their body composition. So approximately 50% penetrance. And that was independent of litters, independent of a founder effect. So it seems to be something that we recurrently see. We don't know exactly what might be the reason for the penetrance issue. Now, what we do know is that when the mice were young, their glucose tolerance was similar to the control mice. All three groups compared together, highly overlapping. And as they get older, there seems to be some split, but not significant. What was really interesting was the insulin tolerance test, where we could see from an early age that regardless of the body weight, liver TMM knockout had a better insulin tolerance curve than the control mice, and that remained in their older years. So loss of hepatic Tmem127 improves insulin sensitivity just by not having Tmem127 in the liver. And we also created an adipose-specific mouse by crossing our flox mouse with adiponectin Cree, and that was, again, a gift from Luke Norton. And both young and older mice maintain an overlapping glucose tolerance test. And this is just fresh from the oven that Subbu did not too long ago. The older adipose-specific knockouts seem to have worse insulin curves. So they trend towards insulin resistance. We don't really have a whole lot of data from these mice yet, but the adipose knockout is not recapitulating what we were seeing with the straight knockout, but the liver specific is. So we have some initial data from the liver knockout. Yeah.
With knockouts in liver and fat, also have differences in size and weight?
The liver does. 50% of them do, right? So 50% of the liver mice are smaller than the flocks, but the other 50% are similar, not bigger. The adipose, they're pretty much the same. There's no difference. Yeah. Yes and no. So body weight-wise, for the CMV knockout, which is what we have the most data from, they are smaller, regardless of gender. The majority of data that we generated were from males. There seems to be less of an insulin-sensitive phenotype and less of a hepatic de novo lipogenesis phenotype with the females, but we haven't really tested them to the same extent that we did the males to tell you exactly how. Well, I'm calling the decreased fatty acid protein levels, yeah. So this is looking at the mRNA levels of the same fatty acid synthesis genes that were reduced on the CMV knockout mouse. And here in blue is the flox control. The green is the liver knockout that does not have a change in size or weight and composition, and the small liver knockout mouse. We call them the big and small. And as you can see here, they don't really mimic the lipogenesis pattern that we were seeing with the CMV straight knockout. However, they do have insulin sensitivity. So if we split the two phenotypes into insulin sensitivity being decoupled from the lipogenesis, then what the liver knockout has in common with the CMV knockout is that they're both insulin sensitive, but they differ with respect to hepatic lipogenesis expression levels. Because of this conundrum, we also wanted to see, so is the liver cell autonomously responsible for the phenotypes that we see in the absence of Tmem127? So we generated CRISPR-Cas9 based Tmem127 knockout in HepT2 cells, which are hepatocarcinoma cells. So we understand some caveats that may come with it because it's not a normal liver cell, it's a cancer cell. But it has been used in many situations as an additional alternative model. So we are using it within that context with all the caveats that we can envision. So the cells are truly knockout, and we look there for insulin signaling and de novo lipogenesis with the models that we had presented for the mouse. So in terms of insulin signaling, we looked at AKT, and we found that the knockout has higher AKT levels, so kind of similar to this CMV straight knockout. And we also found that it has a decrease in the levels of ACC and fatty acid synthase in the knockout compared to the wild type. Now, in this particular model, we were able to show that SREBP1, which is one major master transcription factor regulator of fatty acid synthesis in the liver, is not being processed fully. So what you're seeing here on this Western blot is the unprocessed form, and the processed form that goes to the nucleus is the active form. So you can appreciate that in the knockout, there is more of the unprocessed form relative to the processed form. So potentially in these cells, SREBP processing defect might be one of the elements involved in the reduced fatty acid synthesis protein expression levels. But to a great extent, it kind of gave us a similar idea to what we were seeing in the mouse model. So TMEM deficiency seems to improve insulin signaling and attenuate lipogenesis, which is an interesting combination. And it goes against the conventional wisdom that having more insulin would be lipogenic. So could TMEM deficiency be a way to specifically improve insulin sensitivity without the negativity of having lipogenesis going on? So an interesting idea to at least try to sell and grant. So that brings me to the final portion of the talk, and this is straight from one of the slides that Subbu presented to you not too long ago, just showing what could be a potential relevance of understanding Tmem127 and its deficiency in the context of hepatic fatty acid disease. So fatty liver disease is highly prevalent. It's becoming even more prevalent with the existence of obesity at epidemic levels. And it can progress all the way to hepatocarcinoma under specific progression circumstances. It's very frequent in diabetic patients. So could Tmem127 have a role there? So this is data from Sunil Ahuja. He has a collaborative group where they collect liver biopsies from patients that have fatty acid disease at various different stages. And what I'm showing here is the expression levels of Tmem127 by RNA-seq in a control population compared to different stages of severity of the hepatic disease. And it's highly significant when you compare controls with the higher stages or low stage with high stage. So TMEM is correlated with the severity of fatty acid liver disease in humans. And in these patients, there is data from insulin levels, and it seems to have a very significant also correlation. So Tmem127 levels correlating with Tmem127 mRNA expression in the liver correlating with insulin levels of patients that have fatty acid liver disease, which is quite exciting and may suggest that there is really a translational component to the existence of Tmem127 levels and development of fatty acid liver disease. So this is a summary of what I spoke. Liver-specific TMEM knockout recapitulates the insulin sensitivity of the straight knockout to a great degree and supports the liver as being the primary target. And that is not the case for the adipose absence of Tmem127. And it also suggests that these are really the balance between potentially opposing effects between the liver and the adipose tissue is kind of the mirror image of what Richter does to these two tissues based on the in vivo model. So suggesting that, in fact, TMEM might do some of its effects through mTORC2. So I wanted to acknowledge again Subbu, as I promised. He did all of this work. Elon did all the data on the association between TMEM and the lysosomal complex. Shahida is working on the primary tumors and mutations. Zimin does a lot of work on across different projects. These are people who have been in the lab before, Chin, Anchi, and Gao, who did different portions of the work that I presented today. I have many collaborators here who helped us through the process of getting acquainted or reacquainted with metabolic endocrinology and have provided us with reagents and advice, etc. And my funding sources, I'll be happy to answer questions.